Hello, everybody. I'm Doug Martin. I'm a faculty member at Auburn University College of Veterinary Medicine, and I'm the director of the Scott Ritchie Research Center. I have three of my great colleagues here today to talk about our program on gene therapy for neurologic diseases. The specific neurologic diseases that we've collaborated on primarily are called storage diseases. And because many of you will not have heard of storage diseases, I'm going to take just a couple of minutes and introduce them before introducing the collaborators. Storage diseases are a group of disorders, roughly 50 or more, that result from lack of a specific enzyme. Among the most or the best known storage diseases would be Tay-Sachs disease, Gaucher disease, Pompe disease. If any of you saw the movie Extraordinary Measures with Harrison Ford, that movie was about Pompe disease, which is one of the storage diseases that is in this class. Overall, as a group, the storage diseases are about as common as cystic fibrosis or hemophilia. And so together, they're not all that uncommon, but individually, they are rare diseases. And so we have been working together for about 15 years or so to come up with gene therapy treatments for storage diseases. At the end of the talk or the presentation today, Gavin Corcoran from Gene Therapies will talk a little bit about their work in another more common neurologic disease called Parkinson's disease. And so we'll look forward to seeing that at the end of the presentation. So very few treatments are available for storage diseases. A lot of the storage diseases involve the nervous system, and those are even more difficult to treat. Some of the storage diseases that involve primarily peripheral organs do have FDA-approved therapies right now, but the neurologic storage diseases have been more difficult to treat. So we'll talk about our efforts to make therapies available for those diseases over the coming 45 minutes or so. So let me introduce my three collaborators today, and I'll just actually let them introduce themselves. First, Miguel Estevez from University of Massachusetts Medical School.
Hi, I'm Miguel Estevez, as Doug said. I'm an associate professor in the Department of Neurology, and my lab is actually in the Gene Therapy Center, the Jorge Gene Therapy Center at UMass. And I've been working with Doug for, I don't know, maybe 15 years or so, when we started actually devoting our attention to developing AAV gene therapies for some of these diseases, and we've come a long way since then. So that's my background.
Fantastic. Heather Gray Edwards, also at UMass Medical School.
Hi, I'm Heather. I am an assistant professor in the Department of Radiology, and I'm also a member of the Jorge Gene Therapy Center. I've been here for about 3 years. My research focuses on using large animal models to assess efficacy in gene therapy preclinical studies for clinical application. And I am from Auburn University originally. I was trained there as a postdoc under Doug Martin, and happy to participate. So thank you.
Thanks, Heather. And Gavin Corcoran is at Gene Therapies.
Hi, everybody. Gavin Corcoran, head of R&D at Gene Therapies. I have been at SCIO now for just over 3 years. And for almost all of those 3 years, we've been collaborating with the rest of the folks that you're seeing here. And so excited to be here and to talk about the great progress that's been made with gene therapies for these diseases, starting with what Miguel and Heather and Doug started with, and then moving them into the clinic where we're involved with. So great to be part of the discussion.
Thanks, Gavin. And I'll just mention very quickly somebody that I can't leave out. It's Dr. Cindy Tift at National Institutes of Health, who is running one of the clinical trials. Unfortunately, Cindy has scheduling conflict and couldn't be with us today, but I want to mention her because she's been involved with us for at least a decade in getting these programs ready to go. Okay, so we talked a little bit about storage diseases and what those are, and because there's a varied audience at this meeting, I thought it might be good to spend just a minute or two talking about what gene therapy is. Gene therapy can involve a number of different concepts or approaches, but I thought Miguel truly is a leader in gene therapy around the world, honestly, internationally known, and I thought it would be good for him to spend a couple minutes just describing what gene therapy means in the context of these storage diseases and developing treatments for them.
Well, okay, well, that's quite the introduction. Thank you. I'm not sure my expertise is worldwide, but I'll take the compliment. Thank you. So, you know, the definition of gene therapy, it's actually interesting. I was teaching medical students yesterday, and one of my first slides is actually this definition of gene therapy. What is it exactly? And it has evolved over the years as technologies become available. We all understand, I think, intuitively that these genetic diseases are caused by point mutations, changes in single nucleotides that have, well, obviously catastrophic consequences. And when we started working together, there was really not that many options, and correcting these point mutations was something that, you know, we could foresee in the future happening, but not really being available at that time. Well, this many years later, actually, that's a reality that the technology evolved to the point that that may actually be possible. At the same time, there is another part of the field that has evolved dramatically, which is using small pieces of DNA or RNA to modify expression of genes inside cells, either manipulate it, reduce it, increase it, many different ways. So the field has become much more broadly defined, and I think the definition perhaps is to change any approach that leads to a change in expression of the genes in a cell to achieve a particular therapeutic outcome. And obviously using genetic information, either very, very small in the form of these oligonucleotides, or in the forms much larger as viral vectors to introduce an entire expression elements necessary to actually tell the cell how to make a normal protein. And nowadays, actually, we have the ability, at least in experimental animals and now some experiments in vivo, in patients actually, in clinical trials, showing that we can actually edit the genome itself in a patient. So the progress, of course, has been incredible. And definition becomes broader and broader as the technology becomes available. In the context of these stored diseases that Doug is talking about, you know, the gene therapy itself is based on engineering a viral vector. And, you know, the reason we use viral vectors is simply because viruses have evolved over millions of years to do what they do, which is infect our cells, and if left unattended, obviously cause catastrophic consequences for the patients. As well, we've been living with one such virus for a while. Not that we actually use that for gene therapy, COVID-19. We actually use other viruses that are much less problematic. In fact, they cause no problems whatsoever, but equally well, they deliver genes very efficiently to human cells and mouse cells and so on. So we use those properties of these viruses to introduce all the information necessary to express or re-express the normal protein inside cells. And so what the technological evolution has been since we've started our projects is really that the tools have become much, much more efficient. When we started, we had to do direct injections into the brain because we work on stored diseases that affect the central nervous system. And in the meantime, other investigators discovered new capsids, new adeno-associated virus vectors that actually can be administered through the bloodstream, and they can actually cross the blood-brain barrier, which is actually quite difficult to do, but for some reason these viruses can do it. And this is AV9, which if you look through the internet, you'll find very quickly lots and lots of programs that are based on using this relatively new capsid that has come on the field in about 10 years, actually. 10 years ago, it was published that it could do that. And since then, we have now a product that is approved for one of these neurological diseases called SMA that has transformative results. So the field is moving very rapidly to take advantage of these new entity and others that are being created pretty much almost daily to actually enhance these properties. And so that's what we've been doing, is really taking advantage of new properties to be able to treat patients. So a bit of a long answer to a relatively short question, but I think the answer as the technology progresses becomes a bit more detailed in the definition of what gene therapy actually is.
Yeah, absolutely. I mean, the answer reflects breadth of the field these days, I think. And I hope the medical students were appreciative of the lecture that they got yesterday.
Well, we'll see what they say when they send the scores back of the teacher, I guess.
Well, so the title of our session is Neurological Diseases Bench to Bedside, and this truly has been a bench to bedside collaboration, starting with development of the AAV vectors literally on the bench top and moving through mouse and cat models and other animal models, sheep, to test the effect of the gene therapy vector in animals that mimic the human size and distribution challenges maybe a little more faithfully than mice do. So Heather Gray Edwards is a veterinarian and a PhD and has a very broad background in animal modeling. So I would like to ask her to talk about the step of this project that involved the animal models and how important good animal modeling is as you're developing therapies.
So before I get started talking about the science side of animal modeling, I would like to take a minute to talk about just the way we do animal modeling as opposed to what everybody thinks we do with animal modeling. You want people like us doing animal modeling because we do our level best to give the animals the best quality of life humanly possible. I'm trained by Doug, so everything that I say probably I learned from him. But I will say that we go to no end to make sure that these animals have an incredible quality of life. We go through more laser pointers than we care to admit, or more cat toys than we care to admit, and every cat gets a new cardboard box to tear up every day. We go through amazing lengths. The sheep live outside like normal sheep. They eat grass like normal sheep. They look at birds like normal sheep. So we do this the way it should be done. And so when I'm talking about these animal modeling experiments, I want you to appreciate that we have a sunny window where the cats get to sit on people's laps and get pet and look outside. And it's a good compromise here. So with respect to large animal models, rodents, humans, mice aren't people. I think everybody will accept that. But most of experiments that are done for biomedical in human use are done in mice. And in the case of a lot of these diseases, the mice either don't get a disease at all with the genetic mutation that they carry that's similar to humans, or they get a disease that isn't the same. In addition, a lot of times the mice, they don't live as long, their brains are smaller, their immune systems are different. You can't do things with them like you can do with other animals that's similar like people to really get an idea of what's happening. So what we have done is identified animal models that have the exact same mutations as people do with these diseases. And we have used this to really get a better understanding of how well do the gene therapies work. So nothing makes it into a larger animal until it's shown its ability to make cells better and its ability to make mice better. And then at that point, we test it in animals, and we are able to do things in animals like you do in people. So they can get an MRI, and we can look at their brain without having to hurt them. We can really analyze their blood very, very efficiently. We can look at CSF very efficiently to check for biomarkers. And what we've found when we've done this is that the same things that happen in patients happen in the animals, from the metabolite changes to the MRI changes to blood work changes to all of these things. It's actually quite remarkable that you can go from a cat or a sheep to a human and really be able to correlate this work. And we've done this. We did this in collaboration with Cindy Tift, who's unfortunately not able to join us, but she sees all of these kids and she sees them year after year after year and has all of this huge breadth of data studying these diseases. And when we went back and looked at the animals, we found the same thing, which is really interesting. But what's really cool is after we treat the animals with gene therapy, then we use those same measures to see what changes, so that when it gets to patients, they can expect to see remyelination on MRI. They can expect to see changes in ganglioside levels in different tissues. They can or in CSF or in blood or whatever. And so really the beauty of doing the large animal experiments is that we are predicting, and actually in patients largely in the animals what to expect. And it also, it's really important because you never, in patients, there's a lot of questions you have, like, why are these things changing? And you can't analyze them as efficiently as we can in the animal models. So I feel like it's really important, and it's really helped with the development of this program and the success of these programs as well.
Beautiful. Agree 100%. So proud of Heather. She's had 2 training grants from the NIH to develop this expertise. I know she gets sick of hearing me say that, but she's truly a world's expert in translational research. The next step along the pathway, so we've talked about developing gene therapy on the bench, testing it in animals. The next stage then is to test the gene therapy in clinical trials and move towards hopefully marketing, and that's where Gavin comes in Gene Therapies. Heather, Miguel, and I have a lot of experience on the development side. Gavin has a lot of experience on getting things into clinical trials and hopefully moving them towards commercialization. So I thought I'd ask Gavin to talk about how he thinks about getting preclinical and clinical collaborators together, how they're identified. And I think it's probably never too early to start thinking about clinical trials when those of us who are in the development process are doing that work. So Gavin, what are your thoughts about that?
Sure, thanks, Doug. I think there are a couple of ways to look at this, right? And so the work that Miguel and Heather did with these particular programs when they started should be thought of from the standpoint of the ultimate goal, which is to be able to get this out and onto the market so that patients, lots of patients can benefit from it. So it's about thinking about how you put it all together, right? So having the expertise that they have, not only with how do you make gene therapy, right, which is what Miguel was talking about, and then Heather was talking about how you take kind of making that gene therapy and put it into animal models. The most important thing I think there is, what kind of animal models do you have, right? Because what we'd like to know from a clinical standpoint is the closest animal model to understanding the human disease. And fortunately, we do have naturally occurring diseases in cats and the sheep that Doug and Heather were talking about. So that gives us a really good idea as how these therapies may work in patients once you put them into patients. So I think as far as this collaboration goes, the importance is bringing that expertise to the table, so to speak, right? To be able to really understand everything about the product, about the gene therapy itself, about how it performs in animal models, to help us to design those studies to be able to show to the FDA 2 things. Are these safe? We're not going to hurt patients by giving them to them? And then do they make a difference in the disease course, right? So understanding what that disease course is like gives us at least some idea of the kinds of things we should look at in the patients. So obviously we want to look for safety. As you put this, it's a virus, right? It's a virus that you put into, that we get virus infections. And so this is a virus that's modified, but could that virus actually cause some kind of an inflammatory reaction and so on when you put it into people? So understanding that from an animal standpoint gives us some idea of how we might want to think about it from a human standpoint. So safety is very important. And then obviously our assumptions when you put a gene in that's missing or not working correctly, when you put that gene in, the assumption is it will make a difference in the disease. What we need to understand, though, is when you put that gene in, it doesn't make so much of the protein that it actually causes a safety issue. So really understanding that from an animal standpoint gives us all those important things we need to think about from a human study standpoint. So the animal study really helps us to do 2 things. Make sure that the FDA understands that in the animal models, we didn't see any safety issues, or if there were some safety things, that we know exactly what to monitor in the patients. And then secondly, how do we translate the positive effects that we saw in the animals into the patients? So as we design the clinical trials, it's again a collaboration, right, between us from an expertise standpoint, and then also a collaboration with the FDA. having a discussion. These are experts at the FDA that understand gene therapy, understand these diseases, to really bring a program to them and say, based on the animal models, this is what we want to test from a clinical standpoint. Do you agree that if we're able to show an effect in, brain, in the brain from an MRI standpoint, can we show that in fact the protein that we want to see is increasing as we test the cerebrospinal fluid? Can we see that it's making a difference in the human disease, which is, are the patients, for example, able to walk better or to be able to do things from a muscular standpoint that they wouldn't be able to do it as the disease continues on? So as we look at all of those things, those are the things that we make that collaboration with the FDA, that if we're able to show that, similarly to what was able to be shown in the animal models, if we can do the same thing in humans, then we can agree with the FDA that this is safe, it's effective because it's showing a positive effect on the disease that's clinically meaningful to the patients. Then in that case, we can go ahead and get it approved, get it out into the general population so that it can be used for patients outside of a clinical trial that will appropriately respond to that. So it's really, if you think about it, it's a whole stepwise, as you learn more, it's everybody bringing their expertise to the table. So, what I've spent doing in my career a lot of the time is drug development. And so really understand what those regulatory hurdles might be in order to be able to get the thing on the market. And it's bringing that part of it. Heather and Doug bring it, and Miguel bring a different set of expertise, and it's kind of putting it all together to be able to kind of get it across the finish line of being able to get it to patients. So it's really, it's a I guess to use a cliche, it takes a village. It really does, right? It really takes a village of all of us working together to be able to get the product across the finish line. So that's, I think, is how it kind of all fits together.
Yeah, absolutely. Thanks, Gavin. I'm going to throw the panel a little bit of a curveball here, but I'm going to ask Miguel. Miguel, as we were going through the development process, what do you think, I think we could have started thinking about commercialization and clinical trials earlier on in the process. And do you think it's ever too soon to start thinking about that? Now, we put a lot of effort into talking to different companies along the way, but and fortunately found Gavin and Sayo. But what are your thoughts about thinking about these types of issues early on in the development process?
You can't avoid a lot of delays, that's for sure. So, we took quite some time developing these therapies, and it's a question of an evolving thought of, as scientists, often we want the perfect experiment, right? Which is, make an animal normal, right? And I often struggle a little bit with this, which is what is statistically demonstrable, right, versus what's meaningful to patients. And that's where that is a little difficult, especially when you're talking about lifespan of large animals that, unlike mice, can survive for a very long time, right? And so the question is, where do you define that crossing point that you think it's worthwhile going and doing tests in patients? I mean, there is a minimalistic approach, which you test one of these vectors and say, well, makes protein, go for it, right? And that happens when there's no animal models. And sure, it's the best shot that you can get. And since there's no animal models, you can tell the patients, I suppose, well, sorry, we're not even going to try. Or you can actually try it and learn from the patients, right? I think when we started, and for many years, we were in the camp of let's try to make it perfect as much as we can. I think, at least personally, I've evolved a little more to try to define what are the important characteristics. And I think that's where having early discussions with companies, with experts in the disease, actually, and that's a really important aspect, is really try to find clinical experts in the disease very, very early on. We're lucky because we kind of fell into it with wonderful colleagues that really know these diseases extremely well. So I'm not giving you exactly an answer, yes or no, and very clear answer. There's not a clear answer to this necessarily. I mean, some people consider that an improvement in survival for a couple of months in an animal is sufficient, right? I think part of the problem also, not a problem, but there has to be recognition that animals are just that, especially mice. I mean, mice, there is the big saying that mice lie. Poor things don't lie, but maybe we take too much out of the experiments, right? I think, we all recognize that if everything that we do in mice translated into humans, we probably would have cured every single tumor that is out there, give or take. So, I think sometimes you have to also recognize that the model is only going to take you so far. And that ultimately you have to going to learn it in patients. Really, how do humans respond? Because we may respond worse, or we may actually respond better. So it's a little bit of a hard decision to make there. Where should you do that jump? Now, from these preclinical development to a clinical development path. Certainly working with companies that have a long-term view, right? I mean, we in the lab work towards demonstrating principles. Companies work towards developing products. And those are very different activities with very, very different quality requirements. It's not that in the lab we're, do terrible things that don't care about quality. It's just different. There's another degree that, it's a step up quite a few rungs when you start this development process. And while in lab we use certain techniques to do it because it's simple, as well, we can afford it. They're always small scale, and we don't really think about that step of, okay, how are they actually going to do this? A company is going to have to make this enough for X amount of patients per year for X many years. How do they are going to do this, right? So something that at the time we really didn't have. I guess to think about it too much, because actually when we started, there's not a lot of gene therapy companies. I think in the evolution of the landscape, I think nowadays it's important for one to really start thinking from very early on. Okay, how are we going to make these vectors? How are we going to produce them? What quality controls are going to have in place? And try to think through how the translation process will occur, and not just what's in front of your nose, which is, having a mouse or a cat survive longer, which of course without that, admittedly, if it doesn't work in a small animal, the likelihood of working in a human is pretty low. But that has to, you have to engage your mind a little differently, especially because nowadays there are more paths towards commercialization of AAV therapies, right? At the time when we started, I think there was maybe one company or so, and being interested only in diseases that had lots and lots and lots of patients. That has changed. So I think it behooves all of us, and to the benefit of the patients as well, to engage in that thought process fairly early on.
Yeah, fantastic. And the point about meaningful outcomes to the patients is really good. That's something that both Gavin and Heather, with their clinical training as physicians and veterinarians, have helped us out with a lot. Hey, okay, so this animal's not normal, but its quality of life is much improved with this treatment. So that's a really good point. Gavin, did you have something to add?
I did. Miguel said it really quickly, but I want to make sure manufacturing and making these vectors and making this material is not easy. It's not easy, nor is it cheap. And it takes, he said it very quickly, but trust me, it's not very quick. And so.
Oh, no, it's not.
And so
I skipped over that one really fast.
Exactly, I noticed that. But I think it's really important for folks to realize that sometimes it feels like these things take a very long time to develop, right? And while we understand the urgency of getting there, we've got to do it right. And part of that actually is the manufacture. A very big part of it actually is the manufacturing part of it. So we shouldn't lose sight of the fact that that's a really, really important part, and it's really difficult. It's not something that can just be done. It's something that really takes a lot of work. And so as we're developing the animal models and doing the human studies and so on, there's a huge amount of work that goes on in order to be able to learn how to make it, because there's a kind of learning part of it in making it, and then learning to make it at a big enough scale so that when more patients can get treated, once it gets out into the general population, there's enough, right? And again, it may be easier to do it on a very small scale, But then when you try to go from a small scale to a big scale, it makes it difficult. And my analogy I always tell people is, when you bake a cake for 6 people, that's reasonable. When you try to bake that same cake for 600, it's not quite as easy as you think it is. So I think that's the best way of thinking about it. It's tough, right? It's tough to do. So I think that's a really important part of this whole process is the manufacturing. We shouldn't lose sight of that.
There is one aspect, it's not just the scale, right? It's actually having the right assays to really be absolutely sure. Look, if we make 2 preps of a virus, and the second one is not that great, well, we'll go ahead and make another one, right? And say, well, something went wrong with that, make another one. When it comes to clinical application, you have to absolutely guarantee that different production runs are going to be the same, right? And developing assays to actually ensure that, that is actually really difficult. And each product, I know this sounds silly because each product is different. It requires an all new development that is absolutely individualized to that product and that product alone. And I've seen some of the tests. I was sitting in a meeting with one of the CEO or CIO members that has been with them for a long time, that is involved in the development. And he was just showing me, just briefly, a report of an assay that they developed. The report was like 200 pages for one assay. I'm not even going to disclose how much they spent because it is an endeavor. Let's put it that way, financially and time-wise. Because I remember he telling me also how long it took them to do that, and it really is. When you go into the commercial phase or preparing to a commercial phase, things change so much that I think those in academia have not a clue of what's involved. The degree of confidence you have to have in your tests that you do it test after test after test, and it's reproducible, and that alone is not trivial. So it's a lot of work that goes into this. It really is. It's almost as much work as goes into the discovery, actually.
Absolutely. Those critical pieces, the manufacturing piece, is so important to the overall process. And fortunately, AAV manufacturing is getting more streamlined, but it's still very, very difficult to do consistently. So our team, this is we're talking about manufacturing, and that's sort of at the later stages of the disease or of the development process. Our team came together roughly around 2006, as Miguel said, about 15 years ago. And it involved basically Miguel and I meeting at American Society of Gene Therapy meeting. It was not gene and cell therapy back then. I think it was American Society of Gene Therapy. And we met each other, and both were working on GM1, gangliosidosis, which is another storage disease, and started collaborating then. Shortly after that, I wanted to mention Sofia Pesticinsky, who's one of our long-term supporters, a parent of a child with Tay-Sachs disease who has an attenuated form of Tay-Sachs. So Vera is in her 4th decade now, and amazingly competent and intelligent person. But Sofia had the idea, why don't we bring together all of the experts in Tay-Sachs disease and related diseases and form this consortium of people working together, which was a fabulous idea. So we formed the Tay-Sachs Gene Therapy Consortium in 2007, and we began working on collaborative studies together with the National Tay-Sachs and Allied Diseases Association. So not only a couple of people in academia, but foundations and parents getting involved in the entire development process, and then later the corporate world as well. The third important piece of that puzzle in the early years was Dr. Gray Edwards, who came along wanting to do a postdoc after her veterinary degree. And again, she brought in that clinical perspective that maybe Miguel and I didn't have so much of. So Heather, what are your thoughts about how the rest of the team came together? When you came on board, we had Miguel and me and our labs, and we knew people in the community, but what are the other important pieces of the puzzle that fell into place to get these treatments into clinical trials?
So I will say that the meetings, the family meetings that occur for these diseases are extraordinarily important. One is that it makes it makes us very much so resolved to working on them. These are horrible diseases. These babies suffer and they don't live very long, and these families are bound together in incredibly tight-knit communities, and they're willing to tell you anything and everything about their kids and what their kids go through. And in addition to that, as addicted and as we are to getting these diseases cured and treated, are the physicians that see these patients. So we mentioned Cindy Tiff, but there's Dr. Florian Eichler, who also participates in the clinical trials, and others over the years that have helped. The single most important thing about these programs, if I had to say what it was, is the fact that all of us talk to each other regularly. And we all talk about what we're seeing. And it started out at the beginning where Doug and I would go to these meetings and we would talk to physicians and be like, okay, what does this phenotype look like? What do these animals look like? What do the people look like? Tell me more about what happens to the kids. Tell me more about this. And now that it's in patients, it's the other way around, where they're reaching out to us and being like, okay, what did you see in the animals? Did this happen? Did this happen? Did this happen? And with that is absolutely important. The good news about the storage disease community is everybody's very open and everybody likes to talk about what they're doing and with enthusiasm. And we've made great collaborations that way. Presenting data, we're all very good about presenting and publishing data, and so we get to meet other people that are doing similar things or complementary expertise. So, for example, biomarker discovery, is an important part of clinical translation, something that you can use to show a therapeutic effect, a quantitative measure, and through these interactions with other institutions and other people, we were able to establish some great mass spectrometry based biomarkers. You take some really nerdy chemists and bring them in a room with us, and the next thing you know, we have a really great biomarker that is actually being used in patients now. So really just being open about the data, presenting the data, talking about the data, finding out more about what happens in the patients, comparing that to the animals, going back to the patients, going back to the animals, is I think why we're where we are today.
Agreed. I think the communication part is so essential to all of it. And another thing, we're fortunate in our group that the egos have not gotten in the way. Everybody is working towards the same goal, and whoever's the best person to lead whatever particular portion of the project is underway is the person who goes with it. That's been a tremendous part of the overall process too. What about, so you talked about how bad these diseases are and the kids really are in bad shape, but what are the high points along the road to the clinical trials, Miguel and Heather? What, when you look back to the development process and getting to that first clinical trial, what are the positive aspects?
Oh, hope for sure. I mean, these families, they're our biggest cheerleaders. I mean, there's this, it's a lot of hope and a lot of positive vibes going forward. We've always known what would happen to these kids. It's known what would happen to them without a treatment. But now, for the first time, kids with Tay-Sachs, Sandhoff disease, and GM1 gangliosidosis, we don't know what's going to happen to them. And we have hope that they're going to do a lot better and that they're going to improve. And I think from a positive standpoint, this is as good as it gets. I mean, very excited about these kids, and their families are very excited about what's going to happen with them. And I don't think we're able to divulge any information about what's going on with patients, but definitely, one of the Tay-Sachs was discovered in 1880. We've known about this disease for so long, and now we're actually, doing something about it, and I'm proud of that. And I'm hopeful that it'll help. Miguel, your turn.
Uh, the laws, I don't think we need to describe them. I guess research is full of them. And then you have the highs that really pull you up and through. And um, really, I think it's, knowing the families, knowing the patients, and unfortunately, as long as we've been working, also knowing how many patients have died since we've started. It really gives you some perspective on, what do you think is hard, and the effort that you put into it in the lab. It does give you a lot of perspective, and going every year and renew that perspective is quite useful for sure. I mean, the high point really is, no question about it, get to a clinical trial. Now, whether it works or not, it's what I tell usually parents and the people, that you almost rolling the dice again, because humans really are an entirely different entity, right? And I mean, a clinical trial really is an experiment, right? Follows, many, many years of discovery, making vectors of the highest quality possible. But at the end of the day, it really is an experiment, highly controlled, and we're trying to learn something new. And, getting to that stage that there is hope to help the patients. I mean, there's no question that is the high point of all of it, really. I mean, curing an animal or having a positive impact, it's wonderful, of course, because that's the first step towards where we all want to be. But, even just starting to treat patients to find out whether it will make a difference or not is amazing, really. It's great to get there after a long time, but mostly is that sense that, there is some hope. There is some hope now that they can make a difference, right? So yeah, I agree. That's pretty much it.
Fabulous. Yeah, I agree. We were fortunate enough to be at the first clinical trial, the treatment of the first patient for GM1, and I think that was the day that made it all worthwhile. And several other patients have been treated now. I'll leave that to Gavin, because the SIO is basically in charge of the clinical trials, or in charge of managing the clinical trials. So maybe that's a good segue into talking about the status of the clinical trials.
Absolutely. So yeah, I agree. This is exciting times for us to try to see, whether this is going to work, or how well it's going to work once we give it to patients. So clinical trials are broken into basically two pieces. The first part of it is to understand what the right dose is. That's really important. We get some idea from the animal studies, but as Miguel pointed out, people are different, right? And so we've got to try to figure out what that dose is. So we do, the initial part of it is trying to sort out what the dose is, and giving a smaller dose and a larger dose and a larger dose potentially to be able to understand that. And then the second part of it is, once you found the dose that you think is the best dose, then to do a confirmatory study to be able to make sure that it's safe and effective, that dose, to be able to get it out into the general population once you've agreed with the FDA. So for both the Tay-Sachs, or the GM2 trials, and the GM1 that Doug just talked about, we're still in the phase in which we're trying to understand the right dose. So the GM1 study, we've dosed 5 patients that are so-called type 2 patients, or late infantile juvenile patients. We've dosed 5 of those patients at the low dose, and we've dosed 3 of those patients at the higher dose. And then we've started to dose the infantile patients, which are the very youngest patients that usually have the worst progression of their disease, a very fast progression of their disease. We've started to dose those patients with dosing the first one so far. And so we reported out as a company at the end of last year, really encouraging data on those first patients. These diseases progress relatively slowly in some patients, and so you're not going to see an effect in a couple of days, right? It's going to take longer. Again, what do we learn from the animal models? It takes a while to be able to understand what's going on. Nevertheless, the data that was reported out, the 6-month data reported out at the end of last year, was very, very encouraging. We started to see the protein that we wanted to make, right? That enzyme, we saw the increase in the patient's serum after we'd given it intravenously. So what we need to know now, and we'll be reporting that data out at one of the gene therapy conferences in Europe at the end of October, we're looking forward to being able to show the next set of data in a little bit longer, right? The the year data to be able to show the 12-month data. And so that's really important for us to really try to understand that. So we've got that data, and that's the GM1 study that's further along. Then the GM2 study, or the Tay-Sachs study, got started at the beginning of this year. And so far, we've started with the dosing of those patients. And we're excited to see that we're moving along and dosing those patients. Similar thing, right? Trying to understand the disease first, the dose first by dosing at a low dose to start off with, and then higher and higher. And the reason to do that also is from a safety standpoint, right? To really understand and make sure that if you go to a higher dose, that it's still safe to be able to use that dose to take into that confirmatory study. So that's where we are with both the GM1, GM2 studies. We're excited. We see the same kind of hope for these patients. And as Miguel and Heather pointed out, we get to know the patients and families and so on. Generally, we're going to these family conferences and really getting to know and to understand what's important. I think that's really important, right? As we start to look at these kind of confirmatory studies, what really matters, what's clinically meaningful to those patients and families. And it's really important to talk to them, because we may think what we think is important, that may not be important at all, right? It's really what affects their lives every day, and really trying to understand what's clinically meaningful to those patients. So, exciting times. It's really, really exciting times, and partnering up with these folks and being able to partner up with the family organizations as well and see what we can see, right? As Miguel pointed out, we don't know what's going to happen. That's why we're doing the study, right? We're hopeful. We're very hopeful based on what we know from the animal studies, but, we'll see as we do the studies. But as I said, the first piece of data that came out was very, very hopeful that we'll be able to see something.
Fantastic. So as mentioned, the storage diseases are somewhat rare diseases, and some of our audience may not have heard of Tay-Sachs or GM1 gangliosidosis. Most people will have heard of Parkinson's disease, though, and SIO has a program on Parkinson's disease that's even a little further along. Maybe take a couple of minutes in our last 1 or 2 minutes here and talk about the Parkinson's disease program.
Sure, I'm delighted to talk about that. So Parkinson's disease, as Doug pointed out, sadly is not a rare disease, right? It's a quite common disease, and over 1 million people in the United States have Parkinson's disease. So the Parkinson's disease, the problem that causes Parkinson's disease is a lack of a substance that helps nerves to essentially talk to each other, called dopamine. And that reduction in dopamine, because the cells that are producing the dopamine die, we need to replace dopamine or increase the amount of dopamine. You can take it orally, right? For a while it helps, but then you need to take more and more. And as you take more and more, you get side effects as you have to take more of the medication, and it works less well. So this gene therapy, as opposed to replacing the gene, a gene that's not working, this gene therapy actually has 3 genes that code for the enzymes that make dopamine. And so this is a gene therapy that's injected directly into the brain at the point that you need it to help to create this dopamine, so that patients will be able to benefit from the increased dopamine. So we've had kind of generation 1, in which we dosed 15 patients, and now the second generation, in which we've dosed 6 patients so far, and we've seen a response, right? Parkinson's disease, again, not something that goes quickly, and so we need to observe the patients for longer. And we're doing the same thing, increasing dose to try to understand the right dose, and then we're going to do a confirmatory study in which we can do that in a larger population. So things are moving along really well with that program. Again, the manufacturing, that's a challenge, but we're getting there, and needs to be in large quantities, obviously, because it's not a rare disease. And so things are moving along in the program, and we're excited about that one as well. Again, terrible disease, and if we can help patients that have that, we'd be excited to do so.
Thanks, Gavin. Yeah, it's interesting to me as I was listening. So one of our trials is using an IV intravenous delivery. The Tay-Sachs trial is using both a direct brain injection and injection into the cerebrospinal fluid. And then the Parkinson's disease program is using direct injection into the brain. So all different routes of delivery, basically whatever is needed is what I think we are going to do to make an impact for these diseases. It's been a great session. I want to thank Heather and Miguel and Gavin and Russ Lapari from Health Connect South for letting us talk today. if I can do this without messing up too badly, I'm going to share one image. Oh, image sharing is disabled by the host. He knew I would mess it up. I was just going to show a quick picture of our first patient in the clinical trial for GM1 gangliosidosis. Talked to her mother last week or week before, and she continues to improve, which is a fantastic outcome for all of us. Russ kindly allowed me to share the screen, so hopefully you can see the screen now with JoJo here at treatment on the cover of Auburn Veterinarian magazine, and she's quite a celebrity around Auburn. And this is a picture of JoJo about 9 months to a year after treatment in her red sequined jacket. She is a diva. She really loves picking out her own outfits and is doing quite well. So that's basically the take-home message, I think, for all of us is this is what it's all about. So on behalf of all of the panelists, I'd like to again thank Health Connect South for letting us talk today, and any of us would be happy to answer questions offline, I'm sure, anytime. So thanks for your attention.
Thank you.
Thank you.
Have a good day.